Hey out there, internet land. Welcome back to Father Nature's Unique Botanicals. On today's video, we will be discussing, or we will be answering one of your questions. And the question that I received the most is, why do tissue culture plants look different than traditionally grown or traditionally propagated plants? The answer to that is somaclonal variation. Somaclonal variation is a process that happens during the tissue culture process. And there are two different types of somaclonal variation. The two types are genetic variation and epigenetic variation. Genetic variation is, simply put, the genetical variations of the plant from one phenotype to the next, from one seed to the next seed, from one child to the next child, or from one clone to the next. The parent plant carries a host of different traits, and between its children, all can exhibit different qualities, much like human beings or animals, right? So the epigenetic variations, those are a little bit different. Those happen during the culture process, and most of the causes there are going to be physiological, genetic, or biochemical. And going over each one of those in depth, we'll talk about that. The physiological changes are mostly due to the growth hormones and the plant growth regulators that we use when we're culturing. Things such as endo-3-butyric acid, naphthalene acetic acid, or possibly even the 6-benzylamino that many of you start off using for multiplication. During the culture, they're subjected to different conditions like pH changes, temperature changes, and things of that nature, and those can greatly affect the somoclonal variation, the way that it shows itself from plant to plant. So when we go to the genetic causes, there's a lot of things that can happen, right? A plant can either drop chromosomes or it can pick chromosomes up. And what that'll do is it will manifest itself in a variety of different ways, from just the structure of the plant itself all the way to just the look of the leaf. Much like when we see an albo, right? As you see on the screen now. The biochemical causes there are when you do have something such as an albo, right, that doesn't quite photosynthesize the way that its parent would. It must adapt, and it must change its molecular structure truly in order to actually survive, right? So another one of those would have been the antibiotic resistance that happened over time. When you're in culture, or when a plant is in culture, it's subjected to a lot of things, a lot of antifungals, antimicrobials, different biocides, viricides, and antibiotics. That in turn can do much like it does in a human being, and it can gain a resistance to those types of chemicals and antibiotics. So why would we want to do that? How does it work exactly? How do you induce somoclonal variation, right? Well, when you put the plant into callus formation, when you're trying to grow the callus tissue from the explant, that's generally where you would introduce some sort of mutagen. Now that mutagen may just be a hormone or a plant growth regulator. Whatever chemical or nutrient that you're putting in to manipulate the structure of that plant. Once you go past that process, organogenesis occurs, and that's when you're growing the actual organ that you cloned, right? Whether it be a leaf or a node or shoots or what have you, but actually growing that organ out. That's where you'll start to see these variations start to pop out and manifest themselves. Once you go past that stage and you have the actual regenerated plant, as it's called, when it comes into maturity and you're able to take an explant from that, then you can use those variations to put into perpetuity, right? You can either clone that and keep that material going, or you can further mutate that with another round of mutagens. So what factors actually affect the variation? Well, the genotype of the plant, right? The individual plant that you're using, the source of the X plant and where it came from, whether it be a root or node or leaf cutting that you're using, all of those affect the type of variation that you're going to get and the amount of variation that you get as well. Now, the longer that the plant is in culture and subjected to hormones and plant growth regulators, pH changes, the higher likelihood of the variation that you're going to see. And that's where you come into things like your philodendron pink princess. And then you had the pink princess splash. And a few months later, you have the pink princess galaxy. And now we're on to the full on black cherry, right? They're all the philodendron pink princess. And after being cloned and tissue cultured many, many, many times and subjected to these types of hormone changes, the variation is much higher. Right? The variegation that you're looking at is higher, but you're seeing a wide variety of traits that it exhibits, is what it is. Now, why would you want to do that? Right? What is the purpose of somaclonal variation? Why would you induce it? Well, there's a couple of reasons. Some of it is simply just to produce a new variety of plant, a new hybrid of plant, or to come out with two different ones and be able to breed them so that you have an entirely new plant by seed. Another one is to affect the insect and disease control, right? You want to have plants that grow better under conditions that they weren't normally able to grow in or thrive in. So you help aid that by the genetic manipulation of the plant. Lastly is the antibiotic resistance and the stress resistance of the plant. Insect and disease resistance and just antibody resistance altogether. Again, when it's subjected to high amounts of these hormones and growth regulators and antibiotics, they do develop different qualities within that plant. So there you have it, folks. That is the reason that tissue culture plants look different than a traditionally grown plant. I hope this video was helpful. If it was and you like this type of plant nerdy type stuff, this plant sciencey type videos, go ahead and give us a like and give us a subscribe. Share it with your plant nerd friends, right? Hey, thanks for watching. It's been a great time talking to you. We'll catch you on the next video.